Hello guys, welcome to this series of lectures on animal cell culture. Today we are going to discuss the media that are used in animal cell culture. So first let us answer the question, what are media supply? A media is the most important component of a culture environment and it supplies necessary nutrients, growth factors, some hormones to the cells which are being cultured on the media. Also, it helps in regulating the pH of the culture medium as well as the osmotic pressure of the culture medium. So basically, there are three types of media that are used for animal cell culture. Number one, basal media. Number two, reduced serum media. And number three, serum free media. Now, let us see the first type of media that are used in animal cell culture. We have basal media. So, basal media is such type of media that contain essential and non-essential amino acids, vitamins, inorganic salts, organic compounds, trace elements, everything except the growth factors that are necessary for cell growth. So this type of media consists of all the essential, non-essential amino acids, vitamins, organic, inorganic salts, everything except the growth factors that is necessary for the cell growth. That is why we have to supplement this type of media with serum, which supplies the growth factors that are missing in this type of media. Okay, so what are the examples? We have balanced salt solutions, various types of balanced salt solutions we have. These are the examples of basal media. DMEM, RPMI 1640. So these are the most prominent examples of basal media. DMEM means Dalbecko's modified eagle medium. And RPMI 1640 means Roswell Park Memorial Institute. So it is named after the research institute in which this media was discovered. Now let us see the components of basal media. So basal media, as I have already told you, like other media, it helps in maintaining the pH and the osmolarity, and it provides nutrients and energy source. So the main components of basal media are inorganic salts, which helps in maintaining the osmolarity, as well as these salts regulate membrane potential and provides ions for cell attachment and enzyme cofactors. Apart from that, we give a pH indicator, which is phenol red, which helps in visualizing the pH. So there are various buffers which are also used to maintain the pH, like bicarbonate buffer, an HEPAS buffer, and an energy source, which in this case can be glucose. Now there are various supplements to the basal media, because I have already told you basal media lacks some critical essential factors. That's why we have to supplement it with other factors. Okay, one of the most important factors is serum. Apart from that, we have to provide an amino acid called as the L-glutamine, which is an essential amino acid as it is not synthesized by the cell, and it is an energy source because it provides important precursors to the citric acid cycle, and it also provides some precursors which can be used for protein synthesis. However, the major disadvantage of L-glutamine is that it is unstable in liquid medium. Apart from that, we also provide some non-essential amino acids which can act as energy source and can provide precursors for protein synthesis. Apart from this, they may reduce metabolic burden on the cells. We provide some growth factors also and hormones such as insulin, which stimulate the glucose uptake and utilization and stimulate uptake of amino acids, apart from maintaining the differentiation of cells. We also provide some antibiotics and antimycotics. Why? Because as you know, if you leave open the culture medium, then it provides a good source for growth of bacteria and fungi. Okay, so we only want to grow animal cells. So to stop the growth of bacteria and fungi on the culture medium, we have to provide it with supplement it with some antibiotics and antimycotics. So the most commonly used antibiotic that is used is penicillin. Then we can also use streptomycin also. gentamicin, amphotericin B. So these are the commonly used antibiotics which are supplemented in a cell culture medium. So these types of antibiotics reduce the risk of bacterial and fungal contamination. Alright, but the major disadvantage is that if we use long term of these antibiotics, okay, long term use of these antibiotics can render the animal cell culture medium to be antibiotic resistant. So these are preferably avoided for long term cultures. Only for short term cultures these types of antibiotics are given. The second type of media that we have is reduced serum media. So these types of media are basal media formulations, but they contain some additional nutrients and animal derived factors, because of which they reduce the amount of serum that is needed. So because they reduce the amount of serum that you have to supplement with, so that is why these types of culture media are called reduced serum media, because but these types of media contain some factors, okay, essential factors which reduce the amount of serum that is needed. The third type of media that we have is serum free media. Now these types of media circumvents some issues with using animal sera by replacing serum with appropriate nutritional and hormonal formulations. Okay, so serum free media means you don't have to use serum here. Okay, and what do they use instead of serum? They replace serum with appropriate nutritional and hormonal formulations, and these types of serum free media are can be used for specific cell cultures and cell lines like CHO cell, Chinese hamster ovary, Vero cell line, MDCK cell line, MDBK cell lines, like that. And this makes it one of the most advantaged media. Okay, so when we want to use specific, when you use particular types of media for specific cell lines and cell cultures, so serum free media can be used. So these types of media can be very selective for specific cell types and cell cultures by choosing the appropriate combination of growth factors. Now in general we provide serum, okay, to any type of animal cell culture. Suppose we are working with basal medium. Now the question arises, why do we need to provide serum? We need to provide serum because serum provides these factors: basic nutrients, number 1, number 2, hormones and growth factors, number 3, attachment and spreading, because you know animal mammalian cells are adherent cells, so serum helps in attachment of mammalian cells. Binding proteins, why? Because mammalian cells need some proteins to bind to the extracellular matrix because they are adherent cells. So these types of binding proteins are also provided by the serum. Some protease inhibitors are also provided, because when you are providing some binding proteins you have to inhibit proteases, otherwise they will destroy the binding proteins. And it also helps in maintaining the pH, that is, it also acts as a pH buffer. Apart from this, we try to ensure that the media contains adequate oxygen, preferably 20% oxygen of the atmospheric pressure, and carbon dioxide, which can be used for buffering. And the media should maintain the adequate temperature, which is the normal body temperature, 37 degrees Celsius, along with the adequate humidity should also be there. So these are necessity for proper growth of animal cells. Now how can we visualize the change of pH in animal cells? For that we use a pH indicator, which is most commonly used pH indicator animal cell cultures is the phenol red. As you know, phenol red is yellow in acidic medium, tomato red in neutral pH medium, and red in alkaline pH medium. So by observing the change in color of this phenol red, we can figure out whether the cell culture has become more acidic or basic, or it is remaining neutral. Why? Because sometimes as you culture cells, the cells may secrete some compounds, okay, some factors, some organic acids into the culture medium, and that may make the culture medium acidic or basic. So subsequently the cells may die. So we have to have continuous observance of the cell culture media, whether the cells are secreting some factors, some organic acids, or something which is making the medium harmful for the cells. So we have to have these types of pH indicators which will tell us whether the medium has become acidic, basic, or it is remaining neutral.